Welcome students to this program. Today's lecture is on aneuploidy. Generally, chromosome number of different species remains constant generation after generation. However, rarely certain variations in chromosome number occur spontaneously or can also be induced. Such variations give rise to individuals with different chromosome number. You must here remember that chromosome number or the somatic chromosome number of a species is designated by 2n in diploids as well as in polyploids, whereas the gametic chromosome number of a species is designated by n, or the n means the half of the somatic chromosome number, whereas the basic chromosome number of a species is designated by x. X means one complete genome set or a chromosome set. The X in case of polyploids, for example, will remain constant, whereas the N will change. Say for example, we have hexaploid wheat where X remains 7 and the N is 21. In case of tetraploid wheat, for example, the X will remain same, that is 7, whereas N will be 14. Monoploid is a condition which is designated by the complete one set of chromosomes designated by X, whereas haploidy is a condition which is designated by half of the somatic chromosome number. That means in polyploids the haploid condition is different than in case of diploids. In case of diploids, monoploidy and haploidy condition is same. Heteroploidy, the phenomenon of variation in chromosome number among organisms or species is called as heteroploidy. This variation in chromosome number or heteroploidy is of two types, euploidy and aneuploidy. Euploidy means when whole set of chromosome is involved. Either there is increase in whole set of chromosomes or decrease in whole set of chromosomes. That is, it is related to the X, 2x, 3x, 4x, these are different levels of euploidy. Whereas aneuploidy are the changes which involve one or few chromosomes. There can be either increase of a chromosome or there can be decrease of one or few chromosomes. Say for example, a species with 2n is equal to 14, n is equal to 7. In this species, if there are variations in chromosome number, then 2n is equal to 15 or 2n is equal to 13 instead of 14 are the aneuploid situations, whereas 2n is equal to 7, 14, 21, 28 are different euploid conditions. Now, aneuploidy, which is our basic topic today, is a condition where there is an increase or decrease in of one or few chromosomes from a basic somatic chromosome number. This aneuploidy is of two types, hypoploidy meaning loss of one or two chromosomes. This hypoploidy is further of two types, monosomy, which is means the loss of a single chromosome from the somatic chromosome number, or designated by 2n minus 1 condition, or nullisomy, which is designated by 2n minus 2 condition, that is loss of two chromosomes. Then second one is hyperploidy, which means the gain or increase of one or few chromosomes. Similarly, hyperploidy is also of two types. The trisomy condition designated by 2n plus 1 condition, that means there is an increase of one chromosome from the somatic chromosome number of a species, or tetrasomic condition where there is an increase of two chromosomes from the somatic chromosome number and is designated by 2n plus 2 condition. Now first of all, monosomy. Monosomy, as I said, is a condition where there is a loss of single chromosome or one chromosome from the somatic chromosome number, that is 2n condition. So that is why it is designated as 2n minus 1 condition. Now this chromosome loss, since there are many pairs of chromosomes in a diploid cell, or in an individual, eukaryotic individual, this loss of chromosome can be from any pair. So therefore, the number of monosomics which we can get from a species is equal to the number of pairs of those chromosomes in that species, or in other words, in simple terms, the number of possible monosomics is equal to the haploid or half of the somatic chromosome number of a species. Say for example, in a hexaploid wheat where 2n is equal to 42, the number of possible monosomics can be 21, whereas in another species, for example P, 2n is equal to 14, the number of possible monosomics could be 7. Monosomic, since there is a loss of one chromosome, this chromosome carries many important genes for a species. Therefore, generally monosomics do not survive in case of diploids. However, in case of polyploids, since each chromosome is present several times there, therefore polyploids tolerate such a loss of a single chromosome, and hence monosomics are mostly produced in case of polyploid species, both autopolyploid species as well as in allopolyploid species. Monosomics have been produced in different crop plants. For example, in hexaploid wheat, all 21 monosomics have been produced by E.R. Sears in 1954, whereas in case of Nicotiana tabacum, which has 2n is equal to 48, 24 possible monosomics were produced by Claassen and Cameron in 1944. And finally, in cotton, Gossypium hirsutum, which has 2n is equal to 52, 26 monosomics were reported by J.E. Andresi and his co-workers in 1963 and 1984. Monosomic condition is designated according to the chromosome number which is missing. For example, if in a species chromosome number 5 is missing, one of the partners of chromosome number 5 is missing, then the monosomic condition is designated as monosomy 5, or 2n minus 1 in brackets we indicate 5 there, indicating that chromosome number 5 is missing. Then during meiosis, the monosomics show a univalent at metaphase because that homologous pair lacks one of the chromosomes, and hence an univalent is formed. The gametes which are formed by a monosomic individual are n as well as n minus 1, and fertilization of these gametes result into the formation of monosomic, normal as well as nullisomic progenies. Then second is nullisomy. As far as hypoploidy is concerned, nullisomy is a condition where a pair of chromosome is missing. That means, since there are many pairs of homologous chromosomes in an individual, any of the pair can be missing. So therefore, the possible number of nullisomics in an individual is equal to the number of pairs of those chromosomes, or half of the somatic chromosome number. For example, in case of wheat, which has 2n is equal to 42, 21 nullisomics can be possible because there are 21 pairs of chromosomes, and the organisms which lack a pair of chromosomes, they are called nullisomic organisms. Now, nullisomic is much more dangerous condition than monosomic because now both the chromosomes are missing, and therefore the nullisomic condition does not basically survive in case of diploids. However, in case of polyploids, the nullisomics can survive, and that is why in case of wheat, a complete set of nullisomics has been produced by E.R. Sears. During meiosis, the nullisomic condition is identified by the absence of a bivalent at metaphase stage, because that bivalent is missing in case of nullisomic individual, and the gametes which are produced by such an individual are all n-1 types of gametes, and fertilization between such gametes will restore the nullisomic condition. Then we have hyperploidy, which means increase in one or few chromosomes from the basic set. It has two types, trisomy and tetrasomy. Trisomy is a condition where there is one extra chromosome and designated by 2n plus 1 condition, and this extra chromosome can belong to any of the pairs, and hence here also we can say that the possible number of trisomicus in an individual is equal to the number of chromosome pairs or the half of the chromosome number of the somatic set. Then the organisms carrying an extra chromosome are called trisomicus. In case of barley, for example, 2n is equal to 14, there are 7 trisomicus possible. Trisomicus were first obtained in case of Datura stramonium by A.F. Blacksley and his co-workers. T. Suchuya then reported a complete series of trisomicus in case of barley. Trisomicus, you must also know that, are also famous in human beings, where a number of syndromes are reported because of these trisomy. For example, we have Down's syndrome in humans, we have Klinefelter's syndrome in humans. Then this trisomy condition, where there is an extra chromosome, there are further 3 types of trisomicus depending upon whether whole chromosome or a part of the chromosome is involved in trisomy. For example, we have primary trisomy, in which the extra chromosome is a whole chromosome, that is, it is same as are the other 2 partners in a diploid condition of a homologous pair. Whereas there is secondary trisomy, for example, where the extra chromosome is called an isochromosome, that is, the 2 arms of the chromosome are same. Then there is another type of trisomy condition, that is tertiary trisomy, where the extra chromosome is a product of translocation. That means the 2 partners, the 2 arms of the chromosome belong to 2 different homologous pairs. Trisomy, like monosomy, is also named as per the chromosome number which is here extra. For example, if chromosome number 13 in an individual is extra, the individual is called trisomy 13, or n plus 1, we can show in brackets in the numeral 13 there. During meiosis, for example, as far as these trisomics are concerned, a trivalent is found at metaphase stage in case of diploid individuals, and such primary trisomic individuals produce n and n plus 1 types of gametes, and fertilization between such gametes will result into trisomic normal as well as tetrasomic types of individuals. Whereas at meiosis, secondary trisomic also produces a trivalent at meiosis. However, a tertiary trisomic produces a characteristic pentavalent, because in this case the 2 homologous chromosomes are pairs are involved, and a characteristic pentavalent is formed at metaphase stage. Then next one, tetrasomy condition. It is a condition where whole chromosome pair is extra in an individual, so that a particular pair of chromosome which is extra is present 4 times in a diploid condition, and hence this extra pair of chromosomes can belong to any of the pairs, and therefore the possible number of trisomics in an individual is equal to the number of chromosome pairs in an individual. Trisomicus, unlike monosomicus, they successfully survive in case of diploid individuals, because the extra chromosome carries genes. There can be certain effect on the individual, but since there is no loss of the genes, therefore they successfully survive in case of diploids. Therefore, as far as the genetic studies are concerned, cytogenetic studies are concerned, trisomics are generally produced in case of diploids, whereas monosomics are mostly produced in case of polyploids. Now, there are certain other types of aneuploid situations also. For example, we have double monosomy condition, where in an individual 2 different chromosomes from 2 different homologous pairs are missing, and such an individual is designated by 2n minus 1 condition. Or we have a double trisomy, where 2 different chromosome pairs have an extra pair of chromosome and is designated by 2n plus 1 condition. Similarly, we have another situation known as compensating trisomicus, where one of the chromosome from a pair is replaced by another chromosome, but from different species. So this condition is designated by 2n minus 1 plus 1 condition, and such an individual is called compensating trisomicus. Now, origin and production of trisomic, these different types of aneuploids. As I said, aneuploids are produced spontaneously as well as they can also be induced. As far as their spontaneous origin is concerned, different types of aneuploids are generated by non-disjunction of chromosomes or by the formation of a lagging chromosome. Particularly as far as this non-disjunction is concerned, non-disjunction means at the time of metaphase to anaphase segregation, one of the bivalent goes to the same pole, so that distribution of chromosome does not take place, and therefore one of the gamete produced has n+1 type condition, whereas the another gamete where the chromosome has not reached has n-1 condition. And when such gametes take part in fertilization, different types of aneuploids are produced. Similarly, there is sometimes a lagging chromosome during cell division. Lagging chromosome means a chromosome which remains in the center instead of going to any of the poles, and therefore when it remains in the center it is lost, and the pole which has lost this chromosome carries n-1 gamete, and therefore n-1 gamete could be produced under such situations, and hence fertilization between such gametes will result into monosomic conditions. Then there is another situation where these aneuploids could be produced, for example haploid plants, haploidy. Haploidy is a situation where each chromosome is singly present, and in haploids at metaphase stage all the chromosomes, in case of diploids particularly, they remain in a univalent condition, and at the time of segregation of chromosomes from metaphase to anaphase there is complete irregular segregation. Some poles receive more chromosomes, some receive less chromosomes, and therefore there can be 0 to all distribution between the poles, and different types of gametes are produced lacking 1 chromosome or 2 chromosomes, 3 chromosomes or more chromosomes depending upon the chromosome number of that particular haploid individual, and hence different aneuploid gametes are produced, and fertilization between haploid plants and the normal plants will result into all possible types of aneuploids, into the production of all possible types of aneuploids. Then the best source of aneuploid production is triploid plants. Triploids, you must know that each chromosome is present thrice, and at the time of metaphase stage from each triplet or from each trivalent there is an irregular 2:1 segregation at anaphase, or 1:1, whereas another chromosome is lost, or the whole trivalent goes to one pole. In short, different types of aneuploid gametes are produced in case of triploids, and this is the reason you must remember that all the gametes produced by triploids are defective in nature, and fertilization between such gametes will result into the formation of different types of aneuploids. All possible aneuploids can be produced by fertilization between such gametes, and this is the reason that triploids basically are seedless. Those fruits where we do not need seeds, we make them triploid, so they do not produce seeds because of the formation of defective gametes. Triploids can also be used for crosses with diploid plants in order to generate these aneuploid plants. There are some other types of aneuploid origin where aneuploids can origin are, for example, tetraploid plants, where sometimes there is an irregular segregation at anaphase, and hence n-1 or n+1 gametes are formed, and fertilization results into different types of aneuploid production. Then translocation heterozygotes also, since they have a tetravalent, and segregation of that tetravalent at metaphase to anaphase results into n-1 or n+1 gametes, and therefore aneuploid gametes are formed, and hence fertilization between such gametes results into the formation of aneuploid plants, different types. Then as far as the secondary trisomy is concerned, which I said that the extra chromosome is an isochromosome. This isochromosome is basically formed when they are in a trisomic which has an extra chromosome, or a monosomic which lacks a chromosome. So there is a univalent basically in both the cases sometimes formed, and hence that univalent undergoes a transverse division. that chromosome instead of the equational division. So that transverse division results in the formation of an isochromosome, and hence the gamete later on formed becomes XN plus isochromosome, and fertilization between such a gamete with the normal gamete results into the formation of secondary trisomic, whereas the origin of tertiary trisomic is translocation heterozygote. Translocation heterozygote carries a ring of 4 chromosomes which is a product of 2 homologous pairs, and segregation sometimes takes place in such a manner that there is 3:1 segregation, that is 3 chromosome goes to one pole, so that one of the chromosome is the product of translocation, and hence that gamete which is n plus 1 when fertilizes with a normal gamete results in the formation of a trisomic, but the extra chromosome there is a product of translocation, and hence we call that as tertiary trisomic. Then finally, applications of these aneuploidies. Aneuploidies are very useful in studying the effect of individual chromosomes. For example, we make a plant trisomic where there is an extra chromosome. We can study its effect on various characteristics of organism. We can come to know the genes which are present on that chromosome, what types of these traits are affected by this extra chromosome. So accordingly those genes are present on that extra chromosome. Or we can use monosomic condition, for example, in an individual to study the effect of loss of a chromosome. So we can come to know which characteristics are affected by that loss of chromosome, and hence we can come to know what are the possible effects of loss of that particular chromosome. The most important application of aneuploidy is locating genes on chromosomes by using monosomics and trisomics. The most important technique which is used for this purpose, we call that monosomic or trisomic analysis for locating genes on specific chromosomes. Before the advent of this molecular technologies or molecular tools, recombinant DNA technology, earlier the breeders or the cytogeneticists were using this monosomic and trisomic analysis basically for identification of genes on chromosomes. Then aneuploids are also useful in identifying the chromosomes involved in translocation. Similarly, aneuploidy can also be used for generation of alien substitution lines for studying the effects of additional chromosomes. Then monosomics or trisomics particularly, they have been reported in case of humans, resulting into number of different types of syndromes we find. For example, we see there is a Turner syndrome, we see there is a Klinefelter syndrome. Some of the syndromes that I can mention here are, for example, trisomy 21 is a condition which is found in case of human beings, that is chromosome number 21 is extra, and human beings carrying this extra chromosome, they suffer from a syndrome known as Down syndrome. The important effects of this Down syndrome are that body is short statured and there is mental retardation, abnormal pattern of palm are there, and slanted eyes are there. Then there is a condition which is called trisomy 18, that is called Edwards syndrome, found in case of human beings, which results into mental and physical retardation or extreme muscle tone, and early death even can take place in those individuals having chromosome number 18 extra. Then there is a condition which is called Klinefelter syndrome, found in case of males only. For example, males have generally XY sex chromosomes, but sometimes due to non-disjunction of X chromosome, males receive 2 X chromosomes, so the condition becomes XXY condition, and such males suffer from a syndrome known as Klinefelter syndrome. They have lot of sexual abnormalities, or they produce defective sperms, and there is breast swelling in such individuals. Then there is another very important syndrome particularly found in case of females. We call that Turner syndrome or X0 condition. Normally females in human beings are XX as far as their sex chromosomes are concerned, but there is sometimes missing one chromosome, and such females remain short statured and webbed neck, and they are sexually undeveloped. In short, in conclusion, basically, as far as today's lecture was concerned, we discussed about different types of aneuploidy. Overall, these variations in chromosome number are called heteroploidy, which is of two types: euploidy, that is where there is an increase of whole sets of chromosomes, or aneuploidy, second condition, where there is an increase or decrease in one or few chromosomes. Aneuploidy is further classified into two types: hypoploidy, that is loss of one or few chromosomes, or hyperploidy, that is gain of one or two chromosomes. Hypoploidy is further of two types: monosomy condition, where there is increase in one chromosome and the condition is designated by 2n-1, and hyperploidy, where there is a decrease of increase of one or few chromosomes, for example trisomy and tetrasomy. In case of trisomy, 2n+1 condition is there, that means one chromosome is extra, whereas in case of tetrasomy, 2n+2 condition is there, that is two chromosomes are extra. Then the possible number of these monosomics or trisomics or tetrasomics or nilsomics, the possible number of these aneuploids in an individual is equal to its half of its somatic chromosome number, or the number of chromosome pairs which are found in that individual, in those individuals. Similarly, the possible origins of these different types of aneuploids are, for example, autotriploid plants, or non-disjunction of chromosomes, or translocation heterozygotes, or haploidy, or lagging chromosomes, etc. So they can originate in different ways, or if we want to induce the aneuploidy conditions, then we have to raise triploid plants or haploid plants, which will automatically produce then different types of aneuploids. Aneuploidy results into the most important application of aneuploidy is the location of genes, to find out the location of genes on individual chromosomes in cytogenetic analysis, as well as we can study the effect of loss or a gain of chromosome on various morphological features of an individual. And finally, in case of human beings, aneuploidy results into various types of syndromes or abnormalities like Turner's syndrome, Klinefelter's syndrome, Edwards syndrome, or we have Patau syndrome, etc. This was all about today's lecture on aneuploidy.